In this video, we're going to explain to you how DNA and RNA extraction with magnetic beads works. Magnetic beads are tiny particles that are superparamagnetic, which means that they only display magnetic behavior in the presence of an external magnetic field. This property enables the beads to be separated in suspension. Magnetic beads are made of iron oxides such as magnetite. On the surface of the beads, there are ligands that can reversibly bind nucleic acids under optimized buffer conditions. Of course, the larger the total surface of the magnetic beads, the more nucleic acids can be bound. Some nucleic acid extraction beads bind DNA, some RNA, and others total nucleic acids. Solid-phase nucleic acid extraction, also known as solid-phase nucleic acid isolation, involves a series of steps consisting of cell lysis, binding, washing, and elution when performed with magnetic beads. Cell lysis is the process of breaking down the membrane of your sample cells to release the cell contents, including DNA or RNA molecules. The lysis step can be performed using chemical methods, physical disruption, or a combination of both. The result of the lysis step is a cell lysate. To perform the binding step, magnetic beads and a binding buffer are added to the cell lysate. The sample is then incubated to allow the nucleic acids to specifically bind to the magnetic beads. Following this, the samples are then placed on a magnetic separator or stand to attract the magnetic beads carrying the nucleic acids. Please note that there needs to be a good fit between the sample recipient and the magnetic separator or stand to ensure that all the beads are attracted. By aspirating and discarding the supernatant, most of the unwanted particles, like cell debris, can be removed. Next up is the washing step, where a washing buffer containing ethanol is added to the sample and mixed. Remaining unwanted particles are separated from the bead pellet, and can be eliminated by aspirating and discarding the supernatant. More than one washing step may be needed to remove all the unwanted molecules, depending on your specific protocol and application. After the last washing step, the samples are left on the magnet for a couple of minutes to allow the remaining washing buffer to evaporate. This is necessary because the ethanol in the washing buffer could otherwise inhibit downstream applications. Please note that it is very important to adhere to the drying time indicated in the manufacturer's protocol to avoid over-drying the beads, which can result in cracks and nucleic acids that can't be eluted anymore. Finally, the samples are removed from the magnet and an elution buffer or nuclease-free water is added. The samples are then incubated, and the nucleic acids are released from the magnetic beads. The samples are placed on the magnet again, a bead pellet forms, and the purified nucleic acids can be transferred into a second recipient vessel. That's it. Your nucleic acid samples are now ready for downstream applications. Please note that the nucleic acid extraction workflow described in this video applies to samples like blood, microbes, or tissue that contain intact cells. Magnetic beads can also be used to purify samples that have previously undergone PCR amplification, to prepare them for downstream applications. The main difference in this protocol is that a cell lysis step is not required. This shortened workflow is therefore called nucleic acid purification, because it only involves sample cleanup steps. If you want to learn how Integra can help you with magnetic bead DNA and RNA extraction, please refer to the links in the video description below. Thanks for watching, and if you like this video, don't forget to subscribe to our channel for more.